Okay, welcome everybody. This is the continuation of Chapter 14. This is our first video podcast of the term. What we're going to do is work our way from the brainstem upward towards the cerebral cortex and describe some of the relevant structures in the brainstem, the midbrain, and the forebrain. We'll begin with the medulla oblongata. Let's point out some of the structural landmarks in the medulla. The olives are a prominent bulge that are lateral to each pyramid. Posteriorly, the medulla oblongata holds the tracks of the gracile and cuneate fasciculae of the spinal cord, which continue as a couple of ridges on the posterior side of the medulla. All the nerve fibers that connect the brain to the spinal cord pass through the medulla, and there are four pairs of associated cranial nerves, 9, 10, 11, and 12. Remember that cranial nerve 9 is the glossopharyngeal, the vagus is cranial nerve 10, the accessory is cranial nerve 11, and the hypoglossal is cranial nerve 12. Now, the gracile and cuneate fasciculae are tracks that run up and down the spinal cord. And what you want to remember is that the gracile and cuneate fasciculae are ascending tracks, and they terminate in the gracile and cuneate nuclei, which we're going to see in cross-section in just a minute. They then will cross over to a structure that is medial to these tracks, and it's called the medial lemniscus. And from there, these structures will ascend up to the brain. So remember, when we're talking about these types of tracks, we're talking about signal transmission. So this is going to be primarily white matter, and as a result will feature as its primary structural component myelinated axons, and these are going to be for rapid transmission. Okay, as we move through the medulla, we note that it contains a cardiac center because of the fact that the vagus nerve is associated with it, and the vagus nerve innervates the heart amongst several other viscera in the thoracic and abdominal pelvic cavity. This adjusts the rate and the force of the heartbeat. It also contains a vasomotor center. The vasomotor center essentially is an autonomic output that will control the diameter of the arteries, and when that happens, we control blood pressure, and we do this by exercising commands over the smooth muscle that we find in the muscular tunics of the cardiovascular system, in both the veins and the arteries, in fact. So this adjusts blood vessel diameter. There are also respiratory centers in the medulla oblongata, which control the rate and depth of breathing. And again, this would be primarily in an unconscious state. We have voluntary control over the respiratory center when we're awake. There are also reflex centers centered in the medulla, which are involved in the coughing, sneezing, gagging, swallowing, vomiting, salivating, sweating, and movements of the tongue and head. So these are reflexive responses for the most part, things that we don't put a lot of conscious thought into. We just basically respond to them. Let's take a closer look at the medulla. What you're seeing here is an anterior look at the brainstem, and let's point out a couple of features. Note the pyramids. The pyramids are these baseball bat looking things, these green baseball bats in the front that I'm pointing to. The pyramidal decussations are the areas in the pyramid where nerve tracks actually cross from the ipsilateral to the contralateral side. So these are largely descending tracks that are coming down from the brain, and these pyramidal decussations are where a lot of these fibers will cross. Other fibers will cross more towards the inferior aspect of the spinal cord, but just note that this is a major descending nerve fiber pathway. The olives are lateral to the pyramid, so there's an olive and there's an olive there behind that nerve, okay? That's the hypoglossal nerve, incidentally, so right behind the hypoglossal you'll see the olives. The pyramids contain descending fibers called corticospinal tracts. They're called corticospinal tracts because the entire nerve tract originates in the cerebral cortex and it descends down the spinal column. Eventually, it'll exit the spinal column through a spinal nerve and onwards it'll go to the effector, which is generally a skeletal muscle. This carries motor signals to skeletal muscles. The inferior olivary nuclei, those bulges lateral to the pyramids, are relay centers for signals to the cerebellum. The cerebellum is that small mass of brain tissue posterior to the cerebrum and the brainstem and is involved in, among other things, motor coordination. The reticular formation is a loose network of nuclei that extends throughout the medulla pons and the midbrain. It contains cardiac, vasomotor, and respiratory centers, and if we were to look at a sagittal cut through the brainstem, what you would find is that the reticular formation is essentially anterior to the cuneate and gracile fasciculi and posterior to the pyramids. We're actually going to have a closer look at the reticular formation. It runs from the medulla oblongata through the pons and into the midbrain and serves as a relay station for activation of the thalamus, which we'll get to in a little bit. Okay, let's take another look at the brainstem. Let's work our way up now to the pons. Oh, by the way, before we leave the medulla, let's note the nerves that are associated with it. So the hypoglossal nerve, we've already pointed out, the spinal accessory nerve, 11, and the vagus nerve, 10, and the glossopharyngeal, which is 9. All right, so it's not a surprise that knowing what we know about each of these nerves, the glossopharyngeal is mixed to the pharynx, okay, and the tongue. The vagus nerve controls the heart and some of the other viscera, the spinal accessory as well, and the hypoglossal is also involved in motor control as well as some sensory from the mouth and the face. So it's not a surprise that these functions are associated with the medulla because these nerves originate from this portion of the brainstem. Okay, so let's hop on over to the pons. The pons is this lime green area of the brainstem which is superior to the medulla oblongata. There are several important structural components of the pons. We want to take note of the superior, middle, and inferior cerebellar peduncles. These are tracts of white matter that connect the pons to other portions of the central nervous system. We'll get into exactly what in just a minute. If we were to look at the open space that is deep to the peduncles, what you'd note is that the fourth ventricle occupies this space. And if you remember from our discussion on the ventricles of the brain, this is one of the many places where cerebrospinal fluid is generated. And from the fourth ventricle, it flows through the lateral and median apertures where it bathes the exterior of the brain, and it also flows down the central canal where it acts to remove waste products from the spinal cord as well as provide a homeostatic extracellular environment and also a source of nutrients. This is a posterior-lateral view of the brainstem, so what we're looking at is basically kind of behind and to one side. Note that the medulla oblongata is in dark green, the pons is in light green, and the midbrain here is in this sort of rust-colored um, cast. Okay, superior to the midbrain, we see the beginning of the forebrain, specifically the diencephalon. These two egg-shaped lateral protrusions that mark the transition from the midbrain to the forebrain. Okay, let's talk a little bit more about what the pons does. It's an anterior bulge in the brainstem. It is rostral to the medulla. It contains cerebellar peduncles, which connect the cerebellum to the pons and to the midbrain. It also contains ascending sensory tracts and descending motor tracts. The pathways in and out of the cerebellum are characterized by these peduncles. The superior cerebellar peduncle is the primary pathway for information from the pons to the rest of the central nervous system. The middle cerebellar peduncles are basically brain inputs to the pons, and the inferior cerebellar peduncles are spinal inputs into the pons. Okay. The cranial nerves that are associated with the pons are 5, 6, 7, and 8. With 5 being the trigeminal nerve, 6 being the abducens, 7 being the facial, 8 being the vestibulocochlear. So in order of function, trigeminal is mixed to the face, so it has both motor and sensory roles for the face. The abducens is involved in eye movement, so the movement of the eyeball. The facial nerve is sensory to the face as well as some motor control. And the vestibulocochlear is the nerve of hearing and balance. It has inputs from the cochlea as well as the vestibular apparatus, which are major components of the inner ear, as we're going to find out in probably about 2 weeks. Okay, so let's move on to the midbrain. The midbrain is a short segment of brainstem that connects the hindbrain to the forebrain. It contains the cerebral aqueduct, and remember that the cerebral aqueduct connects the third ventricle to the fourth ventricle. It contains the motor nuclei of 2 cranial nerves that control eye movement, cranial nerve 3, which is the oculomotor, and cranial nerve 4, which is the trochlear. There's several nerves that control eye movement. Remember, the abducens also controls eye movement as well. The tectum is a roof-like part of the midbrain that is posterior to the cerebral aqueduct. It has 4 bulges. These are known as the corpora quadrigemina. They're on the back side of the midbrain. If you remember that picture that we looked at about 2 slides ago, those 4 little bulges that were just underneath the thalamus. Those are the corpora quadrigemina, called quadrigemina because there are 4 of them. The superior colliculi, the top 2, function in visual attention and tracking moving objects as well as in coordinating certain reflexes. And the inferior colliculi receive signals from the inner ear. So again, auditory input. This relays them to other parts of the brain, especially the thalamus. And we're going to find out in a little bit that the thalamus is essentially the gateway to the cerebral cortex. And the cerebral cortex is the seat of higher thought and the origination of motor commands, as well as the termination point of most sensory input as well. If we look at a cross-section of the midbrain, we see several interesting features. Take note of the substantia nigra and the red nucleus, which is part of the tegmentum. Also note the medial lemniscus continues through here, and this gray area is part of the reticular formation, which, as we noted earlier, runs from the medulla oblongata through the pons and all the way up to the midbrain. We also want to make note of the cerebral aqueduct here in the center. It is bordered posteriorly by the tectum. and the central gray matter, which surrounds the cerebral aqueduct as well. So let's talk about some of these features. The cerebral peduncles are two stalks that anchor the cerebrum to the brainstem anterior to the cerebral aqueduct. Okay, so the peduncles here, all right, you can see the tegmentum, all right, and the substantia nigra and the crus, which are all part of the peduncle. There are three main components. The tegmentum contains the red nucleus. It's red because of its high density of blood vessels. Connections go to and from the cerebellum in this portion of the peduncle. This modulates fine motor control. Remember that the cerebellum has a role in coordinating motor activity. What it does essentially is compares the sensory input that it's receiving from the ears and the eyes and the proprioceptors with the actual motion that is being performed by the skeletal muscle, which of course is part of the central nervous system output to the effectors. So if it sees that there is a disagreement between what the sensory input is and what the commands are that are being executed by the motor output, it acts to bring those two inputs into agreement with each other. The substantia nigra is dark gray due to the presence of the black nucleus. This is a motor center that relays inhibitory signals to the thalamus and the basal nuclei and prevents unwanted body movement. Degeneration of this portion of the cerebral peduncle leads to the tremors that we associate with Parkinson's disease. Parkinson's disease is basically a death of neurons in this area that secrete the neurotransmitter dopamine. If you want to see a very good movie about Parkinson's disease and some of the early insights into its etiology, check out Awakenings some night. Very, very interesting true story. The cerebral crus is a bundle of nerve fibers that connect the cerebrum to the pons, and as a result, this is going to be primarily white matter again. These are tracts, nerve tracts. And it carries the corticospinal tracts. So remember that the corticospinal tracts are the tracts that connect the cortex of the cerebrum to the spinal cord. Okay, we start with the first part of the name being the origination of the nerve pathway, and the last part indicating the destination. All right, let's talk a little bit about the reticular formation. It's a loosely organized web of gray matter that runs vertically through all levels of the brainstem. Remember, we talked about the fact that we were going to get a closer look at the reticular formation earlier. Here it is. Okay, see this sort of cylinder of gray matter that is posterior to this bulge in the pons and anterior to the corpora quadrigemina and the tectum and so forth. Okay, what does it do? Well, we already noted the fact that it's located throughout the pons, midbrain, and the medulla. It occupies the space between the white fiber tracts and the brainstem nuclei and has many connections with areas of the cerebrum. There are more than 100 small neural networks without distinct boundaries that are found in the reticular formation. These networks govern somatic motor control. They adjust muscle tension to maintain tone, balance, and posture, especially during body movement. They relay signals from the eyes and the ears to the cerebellum. They integrate visual, auditory, and balance, as well as motion stimuli in motor coordination. So, again, working closely with the cerebellum in order to make sure that what you're doing makes sense with what you're experiencing, right? You want to make sure that if you send a motor command, for instance, to pick up something off of your desk, that what you're seeing happening is in agreement with the motor command that you're sending. And if it's not, the reticular formation and the cerebellum work together to solve that problem. It also contains gaze centers, which allow the eyes to track and fix on objects, as well as central pattern generators, which are neural pools that produce rhythmic signals to the muscles of breathing and swallowing. Now, you probably don't think much about the gaze reflex, but you know that if you move your head, generally you can move your head without your visual field shifting very much because you can keep your eyes fixed on a point in your visual field. And this is an unconscious reflex. It's something we do really without thinking much about it. But if you try, you can overcome this reflex with voluntary input. Try and keep your eyes uh, staring in one particular direction while you move your head. And notice how much your gaze will change and how difficult it would be for you to interpret um, visual images if you didn't have a gaze center and this particular type of reflex. Again, this is uh, coordinated with um, the sensory input that's coming through the optic nerve, as well as the motor movements that are governed by the nerves that control the movement of the eyeball, the abducens and the oculomotor and the abducens, the oculomotor, and the trochlear. Thank you. All right. It also acts in cardiovascular control. It contains cardiac and vasomotor centers uh, that are located primarily in the medulla oblongata within the reticular formation in the medulla oblongata. They also modulate pain sensation. It's one of the routes for pain signals from the lower part of the body, and it is one way that they reach the cortex and you become aware of pain. But it's also the origin of descending analgesic pathways, and these are fibers that act in the spinal cord to block transmission of the pain signal to the brain. Now, if we think about when these types of analgesic fibers might be most active, if you've ever seen an individual who is in shock, this would be somebody who is experiencing massive output of these analgesic pathways in order to block the pain from the brain. It's again a survival mechanism, a reflex, and it's designed to allow us to deal with our situation without focusing on the tremendous pain. Basically, a way to get out of danger. Okay, sleep and consciousness play a central role in our states of consciousness. The reticular formation has no small role in this. It governs functions such as alertness and sleep. Injury can result in irreversible coma. So if we damage the reticular formation, it's possible that you could have several different states of wakefulness and sleep. You could go into a coma, or you could be an individual that never drops off to sleep, and this will play havoc with your overall mental state. It's also involved in habituation. The brain learns to ignore certain repetitive, inconsequential stimuli while remaining sensitive to others, and you probably experience this every day when you get dressed and come to work or school. The first thing that you do when you get dressed is you probably notice the weight of your clothes on your skin and your shoes rubbing against your feet, but as soon as you get on with the business of the day, you don't notice those stimuli as much. The stimuli are still there. It's just that your brain is filtering them out so that you can focus on things that are relevant. If you were not able to do this, if habituation was not properly executed by the reticular formation, your conscious mind would be overwhelmed with all these repetitive, inconsequential stimuli, and you would be unable again to focus on your environment and manipulate conditions to enhance your survival. So again, this is one of those very important adaptive responses that allows us to survive. Very primitive response. if we look at the cerebellum, it looks kind of like hamburger or turkey burger. It's located posterior to the cerebrum. It's the largest part of the hindbrain and the second largest part of the brain as a whole. It consists of right and left cerebellar hemispheres that are connected by the vermis. There's the vermis right there. Okay, so this would be the right hemisphere, this would be the left, and this would be the vermis. Okay, there's an anterior and a posterior lobe as well. It has a cortex of gray matter with folds known as folia and 4 deep nuclei that are deep within the white matter of the cerebellum in each of the 2 hemispheres. It contains greater than half of all our brain's neurons, so there's a lot of neural activity going on in the cerebellum. It's very important. Granule cells and Purkinje cells synapse onto these deep nuclei in the white matter and act as, again, coordinators of motor activity. The white matter that branches deep in the cortex of the cerebellum is called the arbor vitae. So here I am outlining the arbor vitae here, this little thing that looks like a white tree. And again, white matter, so these are myelinated axons. The cerebellar peduncles are 3 pairs of stalks that connect the cerebellum to the brainstem. The inferior peduncles are connected to the medulla. Most input enters the cerebellum through the inferior peduncle. The middle peduncles are connected to the pons. Input from the rest of the brain enters by way of the middle peduncle. And the superior peduncles are connected to the midbrain, and they carry the cerebellar output. So remember that the pons is just anterior to the cerebellum and sort of acts as a house for a lot of these tracts. And we actually addressed these peduncles when we were looking at the pons earlier. These peduncles are thick bundles of nerve fibers that carry signals to and from the cerebellum and act as a method of, again, coordinating motor output with sensory input. The cerebellum monitors muscle contraction and aids in motor coordination. It evaluates sensory input by comparing textures without looking at them. It also governs spatial perception and comprehension of different views of 3-dimensional objects belonging to the same object. It's also a timekeeping center. It allows us to predict movement of objects. For instance, if we see a moving object, it allows us to predict where it'll be in the next few moments. And it helps us predict how much the eyes must move in order to compensate for head movements and remain fixed on an object. So this is, again, part of that gaze reflex. It's involved in hearing. It allows us to distinguish pitch and similar sounding words. It also is involved in planning and scheduling tasks, so it is a timekeeper in many different ways. Lesions in the cerebellum can also result in emotional overreactions and trouble with impulse control. If you look at each of these different functions of the cerebellum, you notice that there's a temporal aspect with all of them. We can start with the top part, the evaluation of sensory input. What we're doing is we're evaluating what we're doing with what we're sensing. Okay, so we're trying to make some sort of sense out of the predicted aspect of our motion, our movement. Okay, the timekeeping center predicts the movement of objects independently of our own bodies, and so we're able to figure out where things are going to be as they move through physical space, and as a result it allows us to adjust, again, our gaze center, using the nerves that control the motion of the eyeball, the oculomotor, the abducens, and the trochlear. The hearing aspect of this you might not think of as really a timekeeping function, but you have to remember that pitch is a characteristic of the frequency of sound. And so when we're able to distinguish different pitches of sounds, what we're actually distinguishing is the differences in the speed with which our eardrum vibrates and allows us to distinguish a high-pitched sound from a low-pitched sound. A high-pitched sound has a very high frequency, and a low-pitched sound has a very low frequency. So again, all of these are temporal in nature in some way, and the planning and scheduling of tasks is obviously um, dependent on a temporal component, right? How long is it going to take for you to get something done? How far in the future is a certain event going to occur? Okay, and again, this is our brain's um, perception of time, the passage of time. So, another very important role for the cerebellum. Okay, let's move on to the most elaborate part of the central nervous system, the forebrain. We want to name the three major components of the diencephalon and describe their location and their function. We want to identify the five lobes of the cerebrum and their function, and identify the three types of tracts in the cerebral white matter. We'll also describe the distinctive cell types and the histological arrangement of the cerebral cortex, and describe the location and functions of the basal nuclei and a series of structures called the limbic system. Okay, the forebrain is what you probably normally think of when you think of a brain. It's that big, wrinkly set of hemispheres distinguished by an outer layer of gray matter and an inner layer composed primarily of white matter with a few isolated spots of gray matter that are called nuclei. And we'll get into what those do in a moment. So the forebrain consists of two parts, the diencephalon, which are those egg-shaped lateral protrusions at the superior aspect of the midbrain, and they basically serve as a connection between the cerebrum and the midbrain. They enclose it, the diencephalon encloses the third ventricle. It's the most rostral part of the brainstem, and it has three embryonic derivatives. Now, when we say embryonic derivatives, what we're talking about is the fact that before you had a nervous system, when you were developing in your mother's womb, you had three primary germ layers, and those germ layers were populations of cells that, when they grew up, were going to give rise to the different organs and organ systems of the body. The three germ layers are the ectoderm, the mesoderm, and the endoderm. The endoderm primarily gives rise to the viscera and the linings of the gut. The mesoderm primarily gives rise to the musculoskeletal system. Some of this is a simplification, but basically this is what these germ layers give rise to. And the ectoderm gives rise to our epithelium and our nervous system. During development, you basically, at some point, um, assume the shape of a flattened hollow disc. Okay, so imagine a hollow frisbee. And on the dorsal side of the frisbee would eventually be what's going to be your dorsal side, your back. And on the ventral side of that frisbee will be what's eventually going to become your viscera. Okay, that would be the endodermal side. On that dorsal portion of the hollow disc, right down the middle of the disc, a streak will develop called the primitive streak, and this is the beginning of the formation of the central nervous system. That primitive streak will begin to invaginate and form a neural tube, and that neural tube will eventually give rise to your spinal cord. While the mid portion of this hollow disc is invaginating, there are also cells along the ridges of the tube before it zips shut that are called neural crest cells, and they give rise to certain structures of the musculoskeletal system as well as the tissues of the peripheral nervous system. So this is how the nervous system begins to be born. At the anterior end of the neural tube, bulges will begin to appear, and these mark the beginning of the formation of the brain. And so there are three primary bulges that eventually give rise to the different portions of the adult brain. And if we were to walk through them from the most primitive state to their most developed state, what you would see are an anterior bulge called the prosencephalon, and a bulge just posterior to that, well, let's say instead of posterior, let's say inferior to that, called the mesencephalon, and then a final bulge called the rhombencephalon. That final bulge, the rhombencephalon, eventually gives rise to the structures of the brainstem. The mesencephalon eventually gives rise to the structures of the midbrain, and the prosencephalon eventually gives rise to the structures of the forebrain, which we're addressing now, and this would be the cerebrum and the diencephalon. So that's what we're talking about when we're talking about embryonic derivatives. Okay, we start out basically as a little bulge or mass of embryonic tissue, and through cues that are present in the extracellular environment, we eventually differentiate and give rise to the structure that you see in the full-grown adult. So the three embryonic derivatives of the diencephalon are the thalamus, the hypothalamus, and the epithalamus. The telencephalon develops into the cerebrum. The cerebrum is the part that you basically you think with. Okay, this is the cerebrum here. I'm outlining it, and notice the diencephalon, which is deep to the cerebrum. Okay. Let's look at the thalamus. The thalamus is an oval mass that lies on each side of the brain at the superior end of the brainstem, just underneath the cerebral hemispheres. It constitutes about 4/5 of the diencephalon. The two thalami are joined medially by a piece of tissue called the intermediate mass, and this runs essentially right through the middle of the third ventricle. It's composed of at least 23 nuclei with 5 major functional groups. Now, when we say nuclei here, we're using a definition of nucleus that's probably the third definition you've heard during the course of anatomy and physiology. The first time you heard the word nucleus, we were referring to the nucleus of an atom, which contains positively charged particles called protons and neutrally charged particles called neutrons. The next time you heard the word nucleus, it was in reference to the smallest component of living matter, which is the cell. The nucleus of the cell contains chromatin, which is the genetic material, or DNA, and the associated proteins, the majority of which are called histones. And this is the seat of information in all cells that are accessed, that are used in order to generate all the proteins that perform the cell's various functions. And now we have the third use of the term nuclei, and when we say nuclei in reference to the nervous system, we're talking about clusters of cells that are thought to carry out a specific function. And so you can see in this slide various multicolored regions of the thalamus. There's a dark green region up here, and a purple region, and a red region, and sort of a yellowish orange region, and 2 blue regions. These are all considered nuclei, and we split them into 5 major functional groups. These are the gateways to the cerebral cortex, and all input to the cerebrum passes by way of these thalamic nuclei. So we come up through the brainstem, and we synapse in the thalamus, and the thalamus will then send the signal the rest of the way on to the cerebral cortex. In the meantime, before it does that, before it lets the signal move on to the cortex, it'll filter this information. The thalamus is involved in motor control by relaying signals from the cerebellum to the cerebrum and providing feedback loops between the cerebral cortex and the basal nuclei. It's also involved in the memory and emotional functions of the limbic system, which we'll get to in a little bit. So notice again these different nuclei, these color-coded nuclei that carry out these different functions. Let's take a look at some of the other structures that are present in the diencephalon. The hypothalamus forms part of the walls and floor of the third ventricle. Hypothalamus is located right there, okay? And this is the thalamus. It extends anteriorly to the optic chiasm, which is where the optic nerves cross from right to left. Remember that the optic nerve is cranial nerve 2. And it extends posteriorly to structures called the mammillary bodies. Each of these mammillary bodies contain 3 mammillary nuclei. They relay signals from the limbic system back to the thalamus. Incidentally, if you are wondering why they're called the mammillary bodies, it's because they have a resemblance to the mammary glands, the breasts, okay, because there's 2 of them and they're paired, and they're basically just opposite the pituitary gland. The pituitary gland is here, and the mammillary bodies would be found somewhere around here, okay? The infibindulum, this little stalk right here, connects the pituitary gland to the hypothalamus. It's a major control center of the autonomic nervous system as well as the endocrine system. It plays an essential role in the homeostatic regulation of all body systems. Functions of the hypothalamic nuclei are as follows. They control hormone secretion. This controls the function of the anterior pituitary. It regulates growth, metabolism, and reproduction, and it also has a role in stress response. Now, you've probably heard health professionals or people in the allied health professions mention the fact that you're as well as you feel, right? Or you're as old as you feel, or you can think yourself sick. And this is addressing something called the mind-body connection, which states that your mental state has a direct control on your physical health. And for the longest time, this was thought to be basically quackery. It wasn't given a lot of weight in the medical community. But we now know, because of the structure of the nervous system, specifically the structure of the brain, the hypothalamus, and the pituitary gland, that there may indeed be a physiochemical basis for the mind-body connection. And it lies in the brain's influence over the function of the pituitary gland. The pituitary gland is essentially the commander-in-chief of the endocrine system, and it governs the functions of other endocrine organs scattered throughout the body. And it does this by secreting hormones that cause other endocrine organs to either upregulate or downregulate their activity. And the state of the brain governs the activity of the pituitary gland. And one of the major functions of the endocrine system is to govern the efficiency of the immune system, how well the immune system responds to infection, how quickly, and how rapidly the wound healing process takes place. And so, because of the connection between the endocrine system and the immune system and the brain and the pituitary gland, we now have a physiochemical basis or a mechanism that might explain what we see in the medical profession as the mind-body connection. So I wanted to touch on that before we talked a little bit more about what the hypothalamus does. Okay, the hypothalamus, as we've already stated, is involved in hormone secretion. It controls the anterior pituitary. It regulates growth, metabolism, reproduction, and stress response. It does this by generating diffusible chemicals called releasing factors that cause the anterior pituitary to secrete a number of different hormones that have wide-ranging effects on the endocrine system. There are also autonomic effects. It's a major integrating center for the autonomic nervous system. It influences heart rate, blood pressure, gastrointestinal secretions, and motility. Remember that the autonomic nervous system is that division of the nervous system over which we have no conscious control, but the functions that it governs are extremely important to our survival. So we have the hypothalamus to thank for coordinating autonomic function. What else? Well, it's involved in thermoregulation. The hypothalamic thermostat monitors body temperature. If we're too hot, it will kick the autonomic nervous system into gear and we will begin to sweat. The skin will redden, and as the sweat evaporates, our body temperature will begin to drop. On the other hand, if we're in a cold environment, what will happen is that blood will be rerouted to our core. Sweat glands will be downregulated. They won't generate as much perspiration, and we'll begin to shiver, and our body temperature will begin to elevate. And this allows us to maintain homeostasis, which is vitally important for our survival, because if our body temperature were to climb too high, what would happen is that the enzymes that carry out the life functions, the enzymes, the structural proteins, and so forth, would begin to denature, and if that were to occur, we would die relatively quickly. On the other hand, if our body temperature were to drop too low, the chemical reactions in the body would begin to slow to the point where we couldn't maintain the life processes. There's an old rule of thumb in chemistry that states, generally speaking, the rate of a chemical reaction decreases by half with every 10-degree drop in temperature. So you can imagine the effect that that would have on all the chemical reactions that take place in the human body. You'd eventually slow and die. So thermoregulation, very important for our survival. It also governs food and water intake. It has a controlling aspect over hunger and satiety centers by monitoring blood glucose and amino acid levels. If they drop, we will experience hunger. If they are elevated, we will experience satiety, or a sense of fullness. So this is one of the ways, again, that we control our fuel intake in order to meet our body's energy needs. It also governs our rhythm of sleep and walking. It controls our circadian rhythm, our sense of 24-hour periodicity, our sense of the passage of time. Our circadian rhythm is essentially our body's response to night and day. It's been shown that irrespective of cues from the environment, our basal metabolic rate and our activity levels tend to rise when the sun is up, and they tend to decline when the sun is down. And we need that periodicity in order to maintain our overall health. When our circadian rhythm is interfered with, when we're getting cues that are in contradistinction to what we believe night and day are, or if we are removed from those cues, sometimes our circadian rhythm can cause us ill effects, right? Because we are in disagreement with what the input is from our environment, and as a result, our mental state can be disrupted. So very important in our sense of the passage of time. The diencephalon also has a role in memory. The mammillary nuclei receive hippocampal signals. The hippocampus is also a very important memory recorder. You could think of it as your body's videotape machine. It records what we experience, and then it plays that information back, and that information gets stored in other portions of the brain. It also has a role in our emotional behavior. It governs our anger, aggression, fear, pleasure, and contentment responses. So again, if we have hypothalamic dysfunction, we might see responses that are not in proportion to our circumstances. We might become emotionally numb or overreact to situations that we experience in our daily lives. Okay, if we wanted to take a look at the cerebrum, you can see that it's split into two hemispheres. This would be the anterior aspect, and this would be the posterior aspect. And This is the longitudinal fissure, which cuts a sagittal crease down the center of the cerebral cortex. The cerebrum is the largest and most conspicuous part of the brain. It's our seat of sensory perception, memory, thought, judgment, and voluntary motor action. The two cerebral hemispheres are divided by a longitudinal fissure, as we just pointed out, and they are connected to each other by a white fibrous tract called the corpus callosum. The gyri and the sulci are the grooves that we see in the surface of the cerebral cortex. They increase the amount of cortex in the cranial cavity, so they provide a large amount of surface area in a very small physical space, which is important for a tissue that is involved in lots of signal transmission, signal evaluation, and all the activities that we associate with neurons. So the more surface area we have, the more complex biochemical processes we can carry out in that relatively tiny space we think of as our cranium. The gyri increase the surface area for information processing capability, and some sulci divide the hemispheres into the 5 lobes, which are named for the cranial bones that overlie them. So again, you can see this superior view of the cerebral cortex. Again, note the gyri and the sulci. Note the longitudinal fissure. Note the central sulcus. That'll come up big later. The central sulcus divides the cerebral cortex into front and back halves. Okay, and here it is winding through here and winding down there. Okay, central sulcus. The parietal lobe underneath the parietal bones, the occipital lobe underneath the occipital bone, and the frontal lobe underneath the frontal bone. If we look at the cerebrum in lateral aspect over here, you can see these the left cerebral cortex. You can see the gyri and the sulci and the central sulcus again. Okay. Note the temporal lobe now that we have a lateral view of the brain. Deep to the temporal lobe, you can see the lobes of the diencephalon. Beneath them, the midbrain, the pons, and the medulla oblongata, and back here, the cerebellum. Okay, so the major landmarks again of the cerebrum and the connecting structures that are part and parcel of the midbrain and the brainstem, so you can get an idea of how all this is put together. Okay, moving on, a little bit more in depth, looking at the cerebrum, we can divide it up into several physical and functional areas. One of the first we want to look at is the frontal lobe. It controls voluntary motor function. It's involved in motivation, foresight, planning, memory, mood, emotion, social judgment, and aggression. The parietal lobe receives and integrates general sensory information, taste, and some visual processing. The occipital lobe is the primary visual center of the brain, noted back here in green. Okay, the parietal here in purple, and the frontal here in sort of the aquamarine color. The temporal lobe contains the areas for hearing, smell, learning, memory, and some aspects of vision and emotion. And the insula, which is revealed here by these two little forceps pulling away at the temporal and frontal lobes of the brain. You can see it underneath, so it's deep to these lobes. It's hidden by other regions, and it's thought to be involved in understanding spoken language, as well as receiving taste and sensory information from visual receptors. So when we look at the organization of the brain, let's go lobe by lobe. The frontal lobe, one of the things you want to note is that this is the higher order thinking area of the brain. This is where we formulate plans, we generate appropriate responses to sensory input, and we perform higher level critical thinking. And it was this lobe of the brain that was routinely operated on in the early days of psychotherapy The operation I'm referring to is the prefrontal lobotomy, where a lot of the neuronal connections in the frontal lobe were severed. This was done to patients who were particularly unruly, and it rendered them very suggestible, and basically they lacked initiative. So this is obviously something that's no longer practiced from a surgical aspect, but as we mentioned in lecture, there are individuals who are inadvertently giving themselves chemical frontal lobotomies as a result of engaging in substance abuse. The parietal lobe posterior to the frontal receives and integrates general sensory information, taste, and some visual processing. And what we're referring to specifically with those roles of the parietal lobe is this area called the postcentral gyrus, which is involved in integrating sensory information in a way that the brain can understand and interpret what all the different inputs are telling the brain to do. The frontal lobe contains the precentral gyrus here, and this is involved in the formulation of plans of action and motor output to effectors. So think of this as the receiving part of the cerebral cortex, and this is the sending part of the cerebral cortex. And we're going to find out that they're actually partitioned even further. There are different regions of the pre and post central gyrus that are responsible for either receiving or producing output to different parts of the body. And we'll look at that a little bit later. The occipital lobe is a primary visual center in the brain. This isn't a surprise because the optic nerve feeds directly into the occipital lobe. If you've ever taken a strong shot to the head, it's entirely possible that your brain can rock back against the skull. The occipital lobe of the brain can then bump against the occipital bone, and that can produce visual disturbance, and that registers what we think of in boxing as a technical knockout. If you've ever had the unfortunate experience to be knocked cold, you'll note that there is indeed visual disturbance associated with that, and it's not surprising considering this is the seat of a lot of the visual input to the brain. The temporal lobe is pretty close to the vestibulocochlear nerve. Your ears are just lateral to the temporal lobe, so it's not surprising that a lot of our interpretation of sound and our receiving of sound input and integrating of sound input happens in the temporal lobe as well. This is where individuals that receive cochlear implants, this is where many of the leads are run in individuals that have nerve deafness, and this can be overcome by the use of the cochlear implants, external microphone, and the input to the brain of actual physical electrical stimuli. This is the kind of stimuli you think of as running through wires in your home, but in this case we stimulate the temporal lobe of the brain, and it's then the job of the individual with the implant to try and make those stimulations into understandable language. And there are areas in the brain, Wernicke's and Broca's area, that are involved with this sort of input in order to take sound and think of it as language. And that's sort of a higher level of sound processing, the idea that you can turn it into language. So the frontal, parietal, occipital, and temporal lobe, it's not surprising that they had these roles. Now again, you might wonder, how is it that we figured all this out? And primarily, most of the work comes from damaged brains, either unfortunate individuals who have had brain damage to these areas and present that specific deficit, or from animal models. And as we've discussed, animal models are only an approximation of what goes on in the human system, so you have to take everything that comes from animal models with a little bit of a grain of salt. But in general, we can state that these functions have at least some relationship to these areas of the brain. Okay, here we're looking at the tracts of cerebral white matter that lie deep to the gray matter. And remember, when we talk about white matter, we're talking about signal transmission, because we're talking about the myelinated axons of the neurons, which are primarily white in color due to the presence of myelin. And you're looking at association, projection, and commissural tracts. And these are basically highways of information either between different parts of the brain or from the brain to the brainstem and subsequently the spinal cord. Most of the volume of the cerebrum is white matter. The glia and the myelinated nerve fibers transmit signals from one region of the cerebrum to another and between the cerebrum and the lower brain centers, and these tracks are diagrammed in figure 1414. The projection tracks that proceed from the cerebral cortex to the brainstem and on down are indicated here in green. The commissural tracks here in yellow. The association tracks in this sagittal section in red. Okay, and so the brain not only is sending information to the brainstem and the spinal cord, but it's also talking to itself. In other words, the right half of the brain needs to know what the left half of the brain is doing. In the event that this doesn't occur, it can lead to conditions such as contralateral neglect syndrome or alien hand, and we discussed each of those in lecture earlier last week. So let's talk a little bit about the different types of tracks. The projection tracks extend vertically between the higher and lower brain and spinal cord. These are centers that carry information between the cerebrum and the rest of the body, as we see in the lower portion, the frontal section, the coronal section through the brain, and these projection tracks are shown here in green. The commissural tracks cross from one cerebral hemisphere through bridges called commissures. Most of them pass through the corpus callosum. This is the corpus callosum. Okay. And they also pass through anterior and posterior commissures that lie either anterior or posterior to the corpus callosum. So this isn't the only level of cross communication between the hemispheres of the brain. It's just one of the major ones. This enables the two sides of the cerebrum to communicate with each other, which is important. The association tracts connect different regions within the same cerebral hemisphere. Long association fibers connect different lobes of a hemisphere to each other, for instance, say the temporal to the occipital, or the parietal to the frontal, and so forth. But short association fibers connect different gyri within a single lobe. So you can see some of these short association tracts up here, and you can see some of these longer association tracts also in sagittal section. Okay. Further analysis of the cerebral cortex shows us that it is made up of layers of nerve cell bodies and their axons and dendrites. The neural integration of the cerebral cortex is carried out in the gray matter of the cerebrum. The gray matter is found in three places: in the cortex and in deeper locations called the basal nuclei, and I'll show you where those are in a moment. We also find them in the limbic system, which are a series of structures that sort of encircle the corpus callosum and as a result also encircle the third ventricle and the thalamus. If we look at the different layers of the cerebral cortex, what we find is that the youngest neurons are on the top, and as we descend into the cerebral cortex, the neurons are older. The way that the brain develops is that the cells that give rise to neural tissue actually climb ladders of glia, ascending through these layers until they reach the outer surface of the cerebral cortex, at which point they have no further place to go, and they will then begin to differentiate and carry out neural function. Newer neurons would then, if they were generated during the formation of the nervous system, climb over the top of those resting on the surface, and as a result the stuff you see on the surface is the most recent brain tissue. The cortex is just a general anatomical term meaning bark or outer covering. This is the layer covering the surface of the hemispheres. It's only 2 to 3 millimeters thick, and it constitutes about 40% of brain mass. It has two types of neurons present in it: stellate cells. These are star-shaped cells that have spheroid somas with dendrites projecting in all directions. You can see them here. These are the stellate cells. Look like little stars. Okay. They also act to receive sensory input and process information on a local level. And the pyramidal cells are tall and conical with an apex towards the brain surface up here. Okay. And a thick dendrite with many branches with small dendritic spines, which include the output neurons of the cerebrum. And so you can see these little pyramidal cells. You can see the cortical surface here. Again, this is dendritic in nature, and these output extensions, these dendritic spines, extend into the deeper layers of the cortex. Okay, so you've got two flavors. You've got large and small pyramidal cells, and what they're doing is they're formulating a plan of action, or they are acting on input from lower brain centers or the spinal cord, and then the generated output will then travel through the layers of the cortex into the white matter. It'll eventually synapse on the thalamus, and then from there it'll be relayed to the rest of the spinal cord. Okay, so you can see again these large and small pyramidal cells. Only neurons that leave the cortex and connect with the other parts of the central nervous system are those that are relaying the information. So again, the stellate cells you can think of as integrators, and these small pyramidals are sort of performing signal processing. Okay, so this is all sort of signal processing stuff, but these large pyramidals are sending information out of the cortex and into the other portions of the nervous system. The neocortex is a 6-layer tissue. That's what you're looking at in this slide. It constitutes 90% of the cortex. It is the most recent evolutionary development in the brain. Okay, so we've talked about the fact that the animals that are lower on the evolutionary scale have brains that look more like brain stems than they do like a human brain, and it's not surprising because the basic survival functions were the first to evolve in order to manage to make it to reproductive age in an environment where there was a lot of natural selection going on. So you had to have reflexes, you had to have also neural control over the visceral functions, the autonomic nervous system and so forth. And all this higher order thinking was something that came along relatively late in evolution, and as a result, the cerebral cortex, the neocortex, is a late comer. Okay, so not only is it the newest part of your own brain, the surface of the neocortex, but it's also the most recent development of the brains of biological organisms on this planet. We've talked about the basal nuclei before. You can see the cerebral cortex, the gray matter out here, but deep to the cerebral cortex and deep to the white matter of the brain are these nuclei, these basal nuclei, and they come in a couple of flavors. The corpus striatum includes the caudate nucleus, the putamen, and the globus pallidus. A subset of that is the lentiform nucleus, named such as, they have that name because they look sort of like a lens, a cross-section of a lens, and it consists of the putamen and the globus pallidus shown here. Okay, and you've got pairs of these. You've got one on each side. Okay, so you can see these deep nuclei. These masses of cerebral gray matter are deep in the white matter and lateral to the thalamus, and they receive input from the substantia nigra of the midbrain and the motor areas of the cortex. Okay, so you've got stuff coming up from underneath, okay, and coming down from on top and hitting these basal nuclei. They then send signals back to both these locations, and specifically the information is designed to control motor activity. There are at least 3 brain centers that form the basal nuclei. We've talked about them already: the caudate nucleus, the putamen, and the globus pallidus. Okay, again, the caudate nucleus here and here, the putamen here and here, and the globus pallidus here and here. The lentiform nucleus is considered to be the putamen and the globus pallidus taken together collectively, while the corpus striatum is the putamen and the caudate nucleus collectively. So if we move on to the limbic system, we've talked about it a little bit already, and you're looking at a sagittal section through the brain, and the area outlined in purple is considered to be the limbic system. And the limbic system are a collection of several structures of the brain that are connected by tracts, and they have a variety of functions with regard to the central nervous system. They're important seats of emotion and learning. Its major components are the cingulate gyrus. So if you look here, this is the cingulate gyrus. Okay, this area here that it's encircling the thalamus. It arches over the top of the corpus callosum, shown here, in the frontal and the parietal lobes. The hippocampus, this part down here, is in the medial temporal lobe, and it's involved in memory. And you might wonder, why is it called the hippocampus? It's named that way because when you dissect it out, it looks just a little bit like a seahorse. That's where it got its name. The amygdala, shown here, is immediately rostral to the hippocampus, and it's involved in formulating emotional responses to stimuli. The limbic system components are connected through a complex loop of tracts that allow for circular patterns of feedback between these different structures, the cingulate gyrus, the hippocampus, the amygdala, and there are other highlighted structures here. You can see these thalamic nuclei. You can see portions here of the orbitofrontal cortex. And again, this is one of those structures because it's a little bit vague in its boundaries. There's a little bit of controversy regarding which structures are strictly speaking part of the limbic system and which are not. What you've got highlighted here in purple, the fornix, the hippocampus, the amygdala, the orbitofrontal cortex, the cingulate gyrus, okay, and the thalamic nuclei, along with these mammillary bodies, are all thought to be part of the limbic system, but you'll get areas of disagreement amongst neuroscientists as to what really belongs here and what doesn't. They have centers for gratification and aversion, and how do we know this? Again, we work with animal models, and we also work with individuals who have damage to some of these structures. And what you find in animal models is that if you ablate the amygdala, you tend to lose fear of situations. You tend not to startle. They've done this with animal models. They've also seen individuals with damaged amygdalas as a result of cancer or surgical resection of these portions of the brain, and they exhibit some of these deficits. The hippocampus is involved in the transformation of short-term memory to long-term memory. Think of it as your brain's videotape recorder. So you record what happens during the day, and then as you sleep, what happens is that information is read from the hippocampus into other areas of the brain involved in memory, and then we're able to then recall that information at a later date. And individuals that have damage to the hippocampus have what's called anterograde amnesia, where they don't remember anything immediate that has happened, like within the last day or two, but everything prior to the damage of this structure they can recall. So again, evidence for the fact that different flavors of memory reside in different physical areas of the brain, and further evidence of the fact that this information gets moved from one part of the brain to the other as it transitions from short-term memory to long-term memory. Okay, the brain is involved in integrative functions as well. Let's talk about some of these. We want to list the types of brain waves and discuss their relationships to mental states. We'll describe the stages of sleep, their relationship to brain waves, and the neural mechanisms of sleep. and will identify the brain regions concerned with consciousness and thought, memory, emotion, sensation, motor control, and language, and we'll discuss the functional differences between the right and left cerebral hemispheres. The higher brain functions we think of as sleep, memory, cognition, emotion, sensation, motor control, and language. These involve interactions between the cerebral cortex, the basal nuclei, the brainstem, and the cerebellum. Brain functions don't have easily defined anatomical boundaries. That's one of the difficulties in studying the brain. That's why it's so hard, if you're a neuroscientist, to pin down a specific function to a specific anatomical structure. We talked about the fact that there is also plasticity, right? There's the ability to utilize other portions of the brain to take over for damage to the brain, and this blurs the boundaries even further. The integrated functions of the brain focus mainly on the cerebrum, but they also involve multiple brain levels. In order to get a gauge on brain activity, we have several methods at our disposal. One of the ones we'll talk about is the electroencephalogram, or EEG. This woman has an EEG, a set of EEG leads attached to the portions of the cranium that are involved in picking up these electrical signals, and you can see some of the different types of brain waves that are generated by this device. It's measuring changes in membrane potential across distance. This is what this device is designed to give you a reading of. This is brain activity in general. They're defined as rhythmic voltages that change resulting from synchronized postsynaptic potentials at the superficial layer of the cerebral cortex, so the part that's closest to the flat bones of the skull. There are 4 types of brain waves distinguished by their amplitude in millivolts and their frequency in hertz. That's cycles per second or waves per second. Persistent absence of brain waves is the common clinical and legal criterion of brain death. Alpha waves, indicated here at the top, have a frequency of about 8 to 13 hertz. They're predominant in awake and resting individuals who have their eyes closed and their minds wandering. They're suppressed when the eyes open or you're performing a specific mental task. Beta waves, shown here, have a frequency of 14 to 30 hertz. You see these in individuals who have their eyes open and are performing mental tasks accentuated during mental activity and sensory stimulation. So you can see a little bit of a relationship between performing a specific task, formulating thoughts, receiving sensory input, and formulating motor output, and the concentration on the task at hand. Here you're relaxed, and here you're concentrating on performing a specific set of tasks, and so you're picking up more activity per second in these neurons that are firing near the surface of the brain in the neocortex, and so it's not surprising that you see an increase in the frequency of these brain waves. Theta waves have a much lower frequency. They're 4 to 7 hertz, shown here. You see them in drowsy or sleeping adults, or if you're awake and under emotional stress. So if you're picking up this sort of brainwave activity when you're awake, you obviously have some sort of emotional input to the activity of your brain, and this is why you see a difference between the theta and the beta waveform. Delta waves, shown down here, have a very low frequency, less than 3.5 hertz, and you see these in adults who are in deep sleep. So we've talked about the fact that the electroencephalogram monitors the surface electrical activity of the brain. It's useful for studying normal brain function, such as sleep and consciousness, and it's involved in the diagnosis of degenerative brain diseases, metabolic abnormalities, and brain tumors. Okay, Now let's talk about another of the integrative functions of the brain. Let's talk about cognition. What is cognition? It's the range of mental processes by which we acquire and use knowledge. Okay, so if you want to put a word to it, it's it's you thinking about stuff. Okay, examples would be sensory perception, thought, reasoning, judgment, memory, imagination, and intuition. The association areas of the cerebral cortex have the above functions. Studies of patients with brain lesions, cancer, stroke, and trauma give us information on what parts of the brain are involved in cognition. The parietal lobe association area is involved in perceiving stimuli. Contralateral neglect syndrome is a result of being unaware of objects on the opposite side of your body, and again, this would come as a result of an interruption between the commissural tracts of the brain, either as a result of disease or surgery. Temporal lobe association areas are involved in identifying stimuli. Agnosia is the inability to recognize, identify, and name familiar objects. This is associated with injuries to the temporal lobe. Prosopagnosia is where a person cannot remember familiar faces, so it's a flavor of agnosia. The frontal lobe association area is involved in planning our responses, and it's also a large component of our personality, as well as our ability to execute appropriate behavior. In individuals that have damage to the frontal lobe, what you find is a change in the personality and in the behavior of the individual after injury to that area of the brain. And if you want a wonderful example of this type of damage, this type of experiment, if you will, you want to refer to page 539 in your book and read about Mr. Phineas Gage and the accident that changed his brain's anatomy and his brain's biochemistry. And this is one of the hallmark medical events that were used to assign different areas of the brain different specific functions. Okay, we've been talking about memory a little bit as well. What is memory? It's information management, the ability to recall stuff that you've previously experienced. It entails learning, which is the acquiring of new information. Memory, which is the information storage and retrieval. Okay, so you acquire a new skill, you then master that new skill, and then when you need to recall that skill again, that information is stored somewhere in the cerebral cortex for retrieval at a later time. Forgetting is the elimination of trivial information, and it's just as important as remembering. We've talked about the fact that individuals with photographic memories, or individuals who are unable to engage in adaptation, have an inability to filter out consequential stimuli from inconsequential stimuli, and as a result it becomes very difficult for them to focus on aspects of their environment that are vital to their survival, because they're so flooded with the noise of sensory input from inconsequential stimuli. Amnesia is the inability to describe past events. Anterograde amnesia is the inability to store new information, and we talked about that with regard to damage to the hippocampus. Retrograde amnesia is where a person cannot recall things known before the injury, and these are physically separate areas in the brain. And we do know that in a normal brain, short-term memory eventually becomes long-term memory, and there must be a mechanism to move that information from one physical location in the brain to another, and this is thought to come from the fact that brain, like all other organ systems in the body is made of living tissue, which is capable of remodeling itself and changing the manner in which it um, synapses, the frequency with which it generates action potentials, and so forth. The hippocampus is thought to be an important memory forming center. It doesn't store memories, but it organizes sensory and cognitive information into a unified long term memory. So, again, think of it as your body's videotape recorder, okay? And think of your brain as the videotape if you want to have a sort of a model to look at how the hippocampus relates to the rest of the brain. Memory consolidation is the process of teaching the cerebral cortex until a long-term memory is established. So, in a sense, your brain goes to school in the formation of memory. So, other parts of the brain learn from the hippocampus, and this is how we start to generate uh, a backlog of information that we can use at a later time. The cerebral cortex is where this takes place. The temporal lobe also has a role in memory consolidation, and as well as the prefrontal cortex. Long-term memories are stored in various areas of the cerebral cortex. Vocabulary and memory of familiar faces are stored in the superior temporal lobe here, and memories of one's plans and social roles are stored in the prefrontal cortex. So again, all important aspects of memory, but different physical locations of the brain involved in storing these different aspects of memory. The cerebellum helps us learn motor skills, and the amygdala is our emotional memory center. It formulates appropriate emotional responses to memories and also to current stimuli. So it has a role both in storing information as well as processing information on an emotional level while it's happening. And again, we know this from damage to these areas of the brain. Okay, we've talked about Phineas Gage. Let's give you a little insight into what happened to this poor gentleman. He was the recipient of an accidental lobotomy. He was a railroad construction worker, and he was the unwelcome recipient of a severe injury that involved the shooting of a metal rod through his cheek out to the top of his head, as pictured in 1420. This was an injury to both the ventromedial region of both frontal lobes. You can see here where the spike exited the brain. He didn't die, which was nothing short of a miracle. It caused an extreme personality change. He became fitful, irreverent, and grossly profane, where before he had been, by all accounts, a perfect gentleman. This was the opposite of his previous personality. Because of these types of events, this event in particular, we were able to assign certain roles to the prefrontal cortex, such as planning, moral judgment, and emotional control. So, again, we assigned to that portion of the brain the deficit in behavior that was a result of the injury. And again, is that a perfect model for mapping out something as complex as the brain and assigning it specific roles for these functions? No. But it's the best approximation we have currently. Okay, let's talk a little bit about emotion. Emotional feelings and memories are interactions between the prefrontal cortex, the thinking part of your brain, and the diencephalon, that portion of the forebrain characterized by structures such as the thalamus. The prefrontal cortex is thought to be the seat of judgment, intent, and control over the expression of your emotions. Feelings come from the hypothalamus and the amygdala. And again, the amygdala and the hypothalamus have a major role in the limbic system. Nuclei generate feelings of fear or love in the limbic system. The amygdala receives input from the sensory systems. It also has a role in food intake, sexual behavior, and it also draws our attention to novel stimuli. One output goes to the hypothalamus, which influences the somatic and visceral motor systems. Our heart will race, our blood pressure will rise, our hair may stand on end, or it may induce something as visceral as vomiting. The other output runs to the prefrontal cortex, and it's important in controlling the expression of our emotions, such as our ability to express love, control anger, or overcome fear. Behavior is shaped by associations between these stimuli, our responses to them, and the reward or punishment that results. Basically, the interplay between our judgment area, our prefrontal cortex, and our emotional area, our limbic system, um, allows us to formulate appropriate behaviors and appropriate responses to stimuli. And as a result, this shapes our personality. Okay, let's talk about sensation. What you're looking at here again is a superior shot of the cerebral cortex. This is the anterior aspect, the posterior aspect. This is the precentral gyrus, this is the central sulcus, and this is the postcentral gyrus. The primary sensory cortex are the sites where sensory input is first received and you become conscious of the stimulus. Association areas near the sensory areas that process and interpret that sensory information are involved in us formulating an appropriate response. The primary visual cortex is bordered by the visual association area. This interprets and makes cognitive sense of visual stimuli. Multimodal association areas receive input from multiple senses and integrate them into an overall perception of our surroundings. And so here you're just looking at the motor cortex, which is the precentral gyrus, and the sensory cortex, which is the postcentral gyrus. For a map of these association areas and for a detailed look at the mapping of the pre and postcentral gyrus, you want to refer to page 541 in your text, figure 1421, which shows you where the association areas are relative to the different lobes of the brain. And for the motor and sensory homunculi, you want to look at page 542, and what it'll show you is which parts of the precentral gyrus, for instance, are involved in controlling different portions of the body, the motor output, okay? And for the postcentral gyrus here in dark purple, it'll indicate for you what portions of the cerebral cortex along this fold of the brain receive input from different parts of the body. And again, that's on page 542. The special senses are limited to the head and employ relatively complex sense organs. The primary cortices and association areas are listed below. We'll go through them. Vision. The primary visual cortex is in the far posterior region of the occipital lobe. And again, refer to page 541 for some of this information. The visual association area is anterior to the occipital lobe, and it occupies all the remaining occipital lobe surface. Much of the inferior temporal lobe deals with facial recognition and other familiar objects. And if you refer to the figure on page 541, what you'll see is that the primary visual cortex is highlighted in yellow and the visual association area is in dark green, and again located in the occipital and the inferior temporal lobes of the brain. If we look at hearing, the primary auditory cortex is in the superior region of the temporal lobe and the insula. That's that lobe of the brain that's deep to the frontal and temporal lobes. The auditory association area is located in the temporal lobe, deep and inferior to the primary auditory cortex. It recognizes spoken words, a familiar piece of music, or a voice on the phone. And again, for us, it's real hard to imagine how you could hear something and not recognize it when you previously didn't have any problem understanding the stimulus. But you can only experience that sort of brain activity if you're working from within the aspect of a damaged brain. For instance, I could give you an example with, let's take visual input. We know that there are individuals who are colorblind. They can't distinguish red from green. And for an individual that can see color, it's very difficult to understand how the world would appear to a colorblind person. If red and green were the same color, what color would that be? It's hard for us to know because we're not operating with the same sensory input. And by the same token, it's very hard for us to explain color to an individual who is colorblind for those particular wavelengths. How would you describe red to somebody who can't distinguish between red and green? It would be difficult to do. Okay, equilibrium are signals for balance and our sense of motion, and they project mainly to the cerebellum and several brainstem nuclei, which are concerned with head and eye movement and visceral function. The association cortex in the roof of the lateral sulcus near the lower end of the central sulcus aids in our establishment of equilibrium. And if we were to look for the equilibrium cortex in the figure on page 541, what you'd want to do is try and find the lateral sulcus near the lower end of the central sulcus. So you can see that kind of running along almost where it points to the primary gustatory cortex, that little bright orange bar down there. But again, it's going to be near the auditory and association area in the primary auditory cortex. So you can see those highlighted in orange. Okay. Taste and smell. Sorry. A little bit more about the association cortex. It's the seat of consciousness of our body movements and our orientation in space. And that's not a surprise because that's the definition of equilibrium. Okay, taste and smell. The gustatory or taste signals are received by the primary gustatory cortex in the inferior end of the postcentral gyrus of the parietal lobe and the anterior region of the insula. Okay, and so again, the insula is not going to be visible in figure 1421, but you can pick up the inferior end of the postcentral gyrus on the parietal lobe, and it's going to be that orange bar. Okay, if you look where it says primary gustatory cortex. The olfactory or smell signals are received by the primary olfactory cortex in the medial surface of the temporal lobe and the inferior surface of the frontal lobe, and that is barely visible in figure 1421. It's the dark green area on the lower left hand side of this sagittal view of the brain, lateral view of the brain. Okay, we've been talking a little bit about the homunculi, so let's look at a homunculus, and it's this strange looking thing shown here. This little man lying over the surface of the brain. You can see here genitalia, toes, leg. There's his hip, there's his trunk, his shoulders, his neck, his little tiny arms, his great big fingers, and his thumb. And then this huge face with eyes, nose, upper lip and so forth, and see how the proportions of the homunculus are sort of out of proportion to our body dimensions. And the reason for this is because the homunculus shown here, this sensory homunculus, has the proportions of this little man uh, directly proportional to the number of inputs that this portion of the body sends to this area of the brain. And so since you have a lot of sensory information coming, for instance, from your tongue, your lips, your face, these areas are overrepresented, whereas sensory information coming from your hip and your back and so forth are underrepresented. So you get a very unusual looking little man. The lateral and medial aspects of the brain here are shown. This would be a coronal section of the right half of the brain. Okay, and the purple area here is the sensory cortex. So remember that this is going to be basically the postcentral gyrus. The sensory homunculus is a diagram of the primary somasthetic cortex, which resembles an upside down sensory map of the contralateral side of the body. It shows receptors in the upper limbs projecting to the superior and medial parts of the gyrus. It shows receptors from the face projecting to the inferior and lateral portions. Somatotropy is this map that you're looking at essentially in this slide. It's a point-to-point correspondence between an area of the body and an area of the central nervous system. So this is what is receiving information from where. The general senses are distributed over the entire body and use simple receptors. Examples would be touch, pressure, stretch, movement, heat, cold, and pain, while the cranial nerves carry general sensations from the head. The ascending tracts bring general sensory information from the rest of the body, while the thalamus processes the input, selectively relaying this input to the postcentral gyrus. The primary somasthetic cortex is the cortex of the postcentral gyrus, and the somasthetic association area is caudal to the gyrus in the roof of the lateral sulcus. Okay, so it's behind it in the roof of the lateral sulcus. So let's talk a little bit more about the general senses. Awareness of stimulation occurs in the primary somasthetic cortex. Making cognitive sense of the stimulation occurs in the somasthetic association area. So this would be the input, okay, right along that ridge, that cortex, that postcentral gyrus. And then what information was received by the cortex, making sense out of it, is the job of the association area. And you'll find this is generally the case with association areas, as they take information and they organize it. Because of decussation, the right postcentral gyrus receives input from the left side of the body and vice versa. Remember that decussation means to cross over from the ipsilateral side to the contralateral side of the body. The sensory homunculus, as we just saw in the previous slide, is an upside-down sensory map of the contralateral side of the body. Somatotropy, again, is the point-for-point correspondence between an area of the body and an area of the central nervous system. And now we finally get to figure 1421, the association areas of the brain and where they lie. Okay, so we can just take a quick tour here. Let's start with the primary motor cortex that lies here in dark blue along the prefrontal gyrus. Okay, the precentral gyrus, sorry, and the primary somatosensory cortex in the postcentral gyrus here in purple. This is from here forward, okay, from the primary motor cortex forward is the frontal lobe of the brain, okay, that I'm outlining here. So in the frontal lobe of the brain, we have motor association areas. What these do is make sense. They formulate a plan of action to be sent to the motor cortex, which then travels down to the spinal cord and to the effectors. Okay, so this is planning what you're going to do, and this is executing it, the brain's method of executing it. Okay, Broca's area is involved in speech, also located in the frontal lobe of the brain. The prefrontal cortex, we've talked about the fact that that's the seat of higher thought. Okay, all our philosophizing, all of our deep thinking comes from this part of the brain. And the olfactory association area is shown down here. Okay, so now you're getting a look at what we were talking about, some of the earlier slides. If we go to the temporal lobe, what you're going to pick up is the auditory association area, and it's not surprising that it's located here because the vestibulocochlear nerve is very closely is located very close to this lobe of the brain. The primary auditory cortex, shown here, is what's going to receive the information from the vestibulocochlear inputs. Okay, so here comes the sound, okay, and then the association area figures out what to do with the sound. What does this sound mean? Are these words? Are these noises? Okay, and then that information may end up getting sent to Wernicke's area back here or Broca's area. Both of these are involved in word recognition and speech. Okay, so again, in comes the input. What is this input? Let's send this input to these areas of the brain and decide either to comprehend it or to speak it or to do whatever with it. Okay, so that's the superior portion of the temporal lobe, okay, sound and language. And other areas of the temporal lobe involve visual association areas located here in green, okay? And this visual association area extends to the occipital lobe, back here in the back of the brain, just superior to the cerebellum, located here. Note that the occipital lobe of the brain contains the visual association areas and the primary visual cortex. So again, vision is coming in from the eyes through the optic nerve to the primary visual cortex. The visual association area decides what this might be that you're looking at, okay? And then other areas of the brain tie it to perhaps stored memory, okay? Or begin to formulate some idea of what the image is or what the image means. Okay, so that's the temporal lobe, the frontal lobe, and the occipital lobe that we've gone through. The parietal lobe is the primary area for somatosensory association. Okay, so input comes to the primary somatosensory cortex from the receptors, and the association area decides what the input might be and what to do about it. Okay, formulates a plan. Okay, and then that information might be sent to the motor cortex or the motor association area, all depending on the nature of the stimulus as it's coming in. Okay, so those are the major areas of the brain and what they do. Gustatory is just your sense of taste, located down here at this inferior end of the postcentral gyrus, this little orange bar. Okay, let's talk about motor control for a little bit. The intention to control a muscle, to contract it, begins in the motor association area of the frontal lobe. We formulate a plan of action in the association area, and it's going to be the motor cortex that's going to actually carry out the plan. This is where we plan our behavior and where neurons compile a program for degree and sequence of muscle contraction required for the action to occur. The program is then submitted to the neurons of the precentral gyrus, which is the primary motor area. Most posterior gyrus is found in the frontal lobe, as we remember from that map on the previous slide. Neurons send signals to the brainstem and the spinal cord, and this ultimately results in a muscle contraction. Some plan of action is carried out. Okay, the precentral gyrus also exhibits somatotropy. Neurons for toe movement are deep in the longitudinal fissure of the medial side of the gyrus. The summit of the gyrus controls the trunk, the shoulder, and the arm. The inferolateral region controls the facial muscles. This is just a restating of the motor homunculus, which has a distorted look because of the amount of cortex devoted to a given body region being proportional to the muscles and motor units in that body region. The pyramidal cells of the precentral gyrus are called upper motor neurons, and their fibers project caudally. Most of them decussate or cross over in the lower medulla oblongata, in that area called the pyramidal decussation. Remember, the pyramids are those green baseball bats that we saw earlier in the talk. They form the lateral corticospinal tracts on each side of the spinal cord. Remember, it's called a corticospinal tract because it runs from the cerebral cortex to the spinal cord and then eventually to the effectors. In the brainstem or spinal cord, the fibers from the upper motor neurons synapse with the lower motor neurons, whose axons innervate the skeletal muscle, and then we carry out the activity. The basal nuclei and the cerebellum are also important in muscle control because what they do is coordinate your actions with what you're perceiving. In other words, you're intending to do something is what you're doing making sense with what you're perceiving that you're doing, and the cerebellum exercises that control between your sensory input and your motor actions. So without the cerebellum, we'd be a little bit lost. Okay, we wanted to pick up with motor control. This is the central nervous system's output to the effectors. It originates in the motor cortex of the brain, descends through the white matter into the brainstem, and from there to the spinal cord, through the corticospinal tracts to the effectors as they exit now the ventral root of the spinal nerve and are then rerouted to the peripheral nervous system. So let's talk about what happens after the motor cortex of the brain formulates a plan of action that's been assembled by the association areas of the frontal lobe of the brain, the motor association areas. The basal nuclei determine the onset and cessation of intentional movements, such as repetitive hip and shoulder movements that you might see in walking. These are highly practiced learned behaviors that once one carries out these behaviors, you can keep executing them without having to think too much about it. Examples will be writing, typing, or driving a car. The source of the control of the basal nuclei over this type of behavior lies in the feedback circuit from the cerebrum to the basal nuclei to the thalamus and back to the cerebrum. Dyskinesia are movement disorders that are caused by lesions in the basal nuclei, and dyskinesia would be essentially motor activity that is not controlled. If we want to get a working idea of what we mean by dyskinesia, it'd be characterized by abnormal difficulty initiating movement, such as rising from a chair or beginning to walk. Examples might be a slow shuffling walk. You see these kinds of problems in individuals with Parkinson's disease, in which the substantia nigra in the basal nuclei begins to malfunction, begins to die. And so what happens is that smooth, easy movements, which require the excitation of agonists. Agonists are muscle groups that help the movement, and inhibition of their antagonists are not properly adjudicated. And so what happens is that the muscle groups fight each other, and it's very difficult to initiate the fluid motion that we associate with voluntary activity. The cerebellum is also very important in motor coordination. It aids in the learning of motor skills. It maintains muscle tone and posture, and it smooths muscle contractions while coordinating eye and body movement. Finally, it coordinates the motions of different joints with each other. So remember that the cerebellum is an area of the brain in which your sensory input and your motor output are synced up, so to speak. And the cerebellum looks for areas of disagreement between what you're perceiving and what you're doing and acts to coordinate these things. And again, all this stuff happens in milliseconds, so it's not something that you would necessarily be aware of if you were suffering from it or even if you were observing it, unless it was an individual with a complete lack of this type of control. Ataxia is a cerebellar defect in which you have a clumsy, awkward gait, and this has to do with the fact that the cerebellum in certain individuals is unable to coordinate these activities due to the presence of something as severe as a lesion, and it can make these tasks very difficult to perform. So two different types of motor malfunctions, dyskinesias and ataxias. Ataxias associated with cerebellar dysfunction and dyskinesia with defects in the basal nuclei. If we look at the motor homunculus, this is figure 14.2.3b. What you see is something similar to the sensory homunculus, but remember that the post-central gyrus is the sensory portion of the cerebral cortex, while the precentral gyrus, which is what we're looking at, is the motor portion of the gray matter of the brain. This is in the frontal lobe, and what we're looking at in this coronal section through the precentral gyrus are those portions of the cortex that are dedicated to sending motor commands to different body parts. And again, you can see how underrepresented the feet, the legs, the trunk, the back, and so forth are, and how overrepresented the hands, the face, the lips, and the tongue are. And this has to do with the degree of motor control dedicated to each of these different body parts. And so it's not surprising that those body parts over which you have the greatest degree of motor control have been allocated the greatest portion of the prefrontal cortex in the frontal lobe of the brain, because this is representative of how much information it takes to run these different parts of the body. So remember again that the prefrontal cortex is that portion of the brain that carries out the plan of action that's generated by the motor association area, which is also in the frontal lobe of the brain. And again, if you want to refer back to figure 14.21, you can get an idea of the physical locations of some of these areas. Now we come to a very interesting aspect of the brain, and it's cerebral lateralization. It's defined as the difference in the structure and function between the two cerebral hemispheres, the left and the right. Again, we're talking about those portions of the cerebrum that are divided by the central sulcus, the longitudinal fissure here. Okay, you can see it moving right down between the right and left hemispheres. So the two sides of our brain are not created equal. The left hemisphere in the majority of individuals is considered to be the categorical hemisphere, and it's specialized for spoken and written language, as well as sequential and analytical reasoning, such as that used in math and science or in anatomy and physiology. It breaks information into fragments and analyzes it in a linear way, whereas the right hemisphere, this portion, is the representational hemisphere, and it perceives information in a more integrated and holistic way. It's a big picture portion of the brain. It's thought to be the seat of imagination and insight, as well as musical and artistic skill, and it also plays a role in our perception of patterns and spatial relationships, our ability to think and see in three dimensions. It also has a role in the comparison of sights, sounds, smells, and taste. It's highly correlated with handedness. The left hemisphere is the categorical one in 96% of right-handed people, and the right hemisphere is the categorical one in 4% of right-handed people. In left-handed people, the right hemisphere is categorical in 15% of these individuals, and the left hemisphere is categorical in 70% of these individuals. Lateralization develops more with age. Males exhibit more lateralization than females and suffer more functional loss when one hemisphere is damaged. And this is thought to be due in some part to a more extensive set of commissural tracts in female versus male brains. So there's more communication between the two lobes of the brain, at least in terms of sheer number of tracts, in females than in males. And this is primarily in the corpus callosum. Okay, now what we want to do is take a quick tour through the cranial nerves. And what we want to find out are what are the 12 cranial nerves, what are their names and numbers. We want to know them by both sets of identification parameters. We're going to identify where each cranial nerve originates and terminates, and we'll state the functions of each cranial nerve. The brain has to communicate with the rest of the body. Most of the input and output travels by way of the spinal cord, but there are 12 pairs of cranial nerves that come from the base of the brain that exit the cranium through foramina. Remember that foramina is simply a term for a hole in something through which something can pass. It could be a blood vessel, it could be a nerve or a lymphatic, or it could be all three. These lead to muscle and sense organs located mainly in the head and neck. If we look at some of these pathways, most motor fibers of the cranial nerves begin in the nuclei of the brainstem and lead to glands and muscles. Sensory fibers begin in receptors located mainly in the head and neck and lead mainly to the brainstem. Sensory fibers for proprioception may travel to the brain in a different nerve from the motor nerve, so you don't necessarily have to travel back to the brain along the same nerve to which from which the sensory fiber relayed input back to the brain. Proprioception, in case we've forgotten, is our sense of body position in three-dimensional space. So this is extremely important for equilibrium, among other things. Most cranial nerves carry fibers between the brainstem and the ipsilateral receptors and effectors. Remember, ipsilateral means on the same side of. As an example, a lesion in the left brainstem can cause sensory or motor deficits on the same side. The exceptions would be the optic nerve, where half the fibers cross over to the contralateral side, and the trochlear nerve, where all the efferent fibers lead to a muscle of the contralateral eye. So those are unusual exceptions in these cranial nerves, in these pathways. Some cranial nerves are classified as either motor or sensory or mixed, and we're going to find out that some of these classifications of nerves are not entirely accurate. In fact, the only purely sensory nerves, as we're going to discover, are the olfactory and the optic, and the rest are actually mixed because they carry a combination of sensory and motor information, but the degree to which they carry these two flavors of information will vary from nerve to nerve, and we'll address that when we get to each of these nerves piece by piece. The classically classified sensory nerves are 1, 2, and 8. Okay, so that's the olfactory, the optic, and the vestibulocochlear. Now, again, this is using classifications that clinicians have been utilizing for a long, long time. Um, Strictly speaking, cranial nerve 8, in addition to carrying um, information about balance, which is our equilibrium from the vestibular apparatus, and sound from the cochlea, also has some motor outputs back to the cochlea. So, strictly speaking, it's not a purely sensory nerve. But again, these are the classic definitions of these groups of nerves. So, sensory 1, 2, and 8, motor 3, 4, 6, 11, and 12. These stimulate muscle but also contain fibers of proprioception. So, again, a sense of body position um, and muscle tension, muscle tone information is also carried along these nerve fibers. And the mixed nerves are 5, 7, 9, and 10. Their sensory functions are often quite unrelated to their motor function. For instance, the facial nerve, which is cranial nerve 7, has a sensory role in taste and a motor role in facial expression. So let's start with cranial nerve 1. And this is just what you're looking at here is an inferior view of the brain. Um, let's make it clear what we're seeing. Okay, there's the medulla oblongata. Okay, there's the pons, and the midbrain is going to be tucked up under here. These are the cerebral hemispheres. And then if we wanted to look at each of these nerves in their actual position, the way that these nerves are numbered is from rostral to caudal. So olfactory nerve, cranial nerve 1 being the most rostral, okay, and the hypoglossal nerve, cranial nerve 12, being the most caudal. So if you look at figure 1427, you can see the locations of these nerves, how they're numbered, the olfactory, the optic, the oculomotor, the trochlear, the trigeminal, the abducens, the facial nerve, the vestibulocochlear nerve, the glossopharyngeal nerve, the vagus nerve here, the hypoglossal nerve, and the spinal accessory nerve. And this is an actual picture of brain tissue here on the right, showing you the locations of these nerves as well. But they're easier, obviously, to see in the color-coded figure on the left-hand side. So let's go nerve by nerve. The olfactory nerve, cranial nerve 1, is involved in our sense of smell. It is a strictly sensory nerve. Damage to it causes an impaired sense of smell. You can see the olfactory bulb and the olfactory tract. This is the cribriform plate of the ethmoid bone, and these are fascicles of tissue from olfactory nerve 1 that descend into the nasal mucosa. The olfactory nerve is special because it is the shortest nerve route from sensory to brain input by far. The other nerves travel quite a much longer distance to get back to the brain. But the other interesting aspect of the olfactory nerve is that it is one of the few central nervous system structures which contains neurons that have to be continuously regenerated because they are damaged as a result of their exposure to the external environment. This is actually a piece of your brain that hangs out in the open air every day and has to constantly be regenerated. The neurons that populate the olfactory bulb are actually born near the occipital lobe of the brain, and they migrate to the olfactory bulb in a path called the rostral migratory stream. So they actually move through the brain tissue on a daily basis. Once they reach the olfactory bulb, they stop, they differentiate, and they begin to carry out their sensory function. So this is one of the more unusual central nervous system structures in the sense that these neurons do regenerate on a daily basis, whereas the majority of central nervous system neuron populations are incapable of being regenerated once they've been damaged. Um, the nasal mucosa is essentially the housing for these fascicles of the olfactory nerve, and in order for odor to be detected, the molecule has to be dissolved in air, and as a result, the receptors can then bind to the odorants, producing a stimulation that goes back to the olfactory cortex of the brain, and we then are aware of odor. Okay, the optic nerve provides our sense of vision. Damage to it causes blindness in part or all of the visual field, and you can see that we have, again, as with the olfactory nerve, two of these, one on the right and one on the left, but the olfactory nerve is unique in that it sends fibers to both the contralateral and ipsilateral side of the brain, and this allows us the luxury of stereoscopic vision because each portion of the occipital lobe receives information from each portion of the visual field, and this is what provides our sense of depth. This gives us the ability to see in 3 dimensions so that we can judge things like distance. And again, this is a purely sensory nerve, one of only 2 by a strict definition. So notice that it is caudal to the olfactory nerve, and that the optic chiasm here lies essentially just anterior to the pituitary gland. Just a few anatomical markers for you to familiarize yourself with the tracks of this particular nerve. The oculomotor nerve is involved in eye movement. It controls muscles that turn the eyeball up, down, and medially, as well as controlling the iris, lens, and the upper eyelid. You can see, okay, this is again cranial nerve 3. You can see its passage essentially from the superior aspect of the pons, okay, between the pons and the midbrain. The superior and inferior branches exit through the superior orbital fissure in the orbital cavity, and we are able to innervate muscles that control eyeball movement as well as iris diameter and the tension on the lens and the upper eyelid. Okay, so this is one of 3 nerves that we're going to see that control eye movement. Damage to it causes a drooping eyelid, a dilated pupil, double vision, difficulty focusing, and the inability to move the eye in certain directions. The trochlear nerve, cranial nerve 4, is another nerve involved in eye movement. It innervates the superior oblique muscle. Damage causes double vision and the inability to rotate the eye inferolaterally, so downward and to the lateral side. And again, we've talked about the oculomotor and the trochlear nerve, 2 cranial nerves that control eye movement. What is it about them that is sensory? What's sensory about them is, again, muscle spindle information that's coming from these muscles, which are skeletal muscles, which control eye movement. So the sensory role is minimal, okay, but it is there. So strictly speaking, this is a mixed nerve as well. Okay, the trigeminal nerve is the largest of the cranial nerves. It's the most important sensory nerve from the face. It forks into 3 divisions. The ophthalmic division, or V1 branch, is the sensory division. The maxillary division, or V2 branch, is also a sensory division. And the mandibular division, or V3, is the mixed division. And so you can see these 3 huge branches of the trigeminal nerve, and they basically exit through foramina in the sphenoid bone. And again, this is a paired nerve. You've got one on each side. And you can see the distribution of sensory fibers for each of these divisions. There's V1, V2, and V3 in this map of the dermatomes of the head. And you can also see the muscles innervated by the mandibular branch, the temporalis, the lateral and medial pterygoids, the masseter, and the anterior belly of the digastric. So this is one of those muscles, one of those nerves that we use when we're chewing stuff. Okay. Incidentally, for any of you who have ever experienced ulcers in the mouth, these are often the result of viral infections that end up being housed in the ganglia, the trigeminal nerves, after an initial exposure, and these viruses will erupt forth in the oral mucosa upon emotional stress or changes in diet. So those are those open sores that people sometimes get in the mucosa of their mouth. It's a viral disease. Antibiotics will not treat it. You can cauterize the wound using alum, which is painful but effective. But the only way to cause the eruption to heal is to remove the source of stress, and then the virus goes latent again. So just like the chicken pox, this is another one of those viruses that can house itself in the central nervous system and the peripheral nervous system, where drugs are difficult. It's difficult to get drugs to treat entities that live in those tissue types because, again, they don't tend to divide and they don't tend to repair. Okay, the abducens nerve is another, it's the final nerve that controls eye movement. It innervates the lateral rectus muscle, and it exits between the border of the pons and the medulla oblongata through the superior orbital fissure. It provides eye movement. Damage results in an inability to rotate the eye laterally, and at rest the eye rotates medially if the abducens nerve is damaged. And again, this is a primarily motor nerve, but there is sensory information coming from the muscle spindles of this skeletal muscle. So technically it is a mixed nerve, but it is, under the classical definitions, considered to be primarily a motor nerve. So all three of the eye movement nerves are technically mixed, but they're classified as motor nerves by the strict definition. So those would be the abducens, the trigeminal, and the oculomotor. Okay, the facial nerve has a motor aspect. It's the major motor nerve of the facial muscles. It aids in facial expression. It innervates the salivary glands and tear, nasal, and palatine glands as well. The sensory aspect are the taste receptors on the anterior two-thirds of the tongue. Damage produces sagging facial muscles and a disturbed sense of taste, where you don't pick up sensations of sweet or salty. When we do the chapter on the special senses, we're going to talk about taste, and we'll note the fact that the taste buds for different taste sensations are located in physically distinct regions on the tongue. And obviously, from our information on the facial nerve, cranial nerve 7, we can note that the anterior two-thirds have some role in the sense of sweet and salty taste. Okay, the vestibulocochlear nerve, again, this is one of those that was classically grouped as a primarily sensory nerve, but again, it is mixed because it also sends motor commands to the cochlea for cochlear tuning. So it's primarily sensory, but there is some motor output. It's our nerve of hearing and equilibrium. Damage produces deafness, dizziness, nausea, loss of balance, and nystagmus, which is an involuntary rhythmic oscillation of the eyes from side to side. And the reason for this, again, is because of a loss of equilibrium, which comes primarily from these structures called the semicircular canals, which are a part of a larger structure, which I'm sort of circling here, which is called the vestibular apparatus, which gives us our sense of balance. It tells us up from down and left from right, and it provides us with our sense of both dynamic and static equilibrium. That would be our sense of body in space when we're moving and our sense of our body in space when we're standing still. The glossopharyngeal nerve, cranial nerve 9, is involved in swallowing, salivation, gagging, and control of blood pressure and respiration. Sensations to the glossopharyngeal nerve come from the posterior third of the tongue. Damage results in loss of the bitter and sour tastes and an impaired ability to swallow. So we can conclude from this information that the taste buds involved in the bitter and sour sensations are located on the posterior third of the tongue. Okay, the vagus nerve. The vagus nerve is an unusual nerve in that it innervates organs distant from the cranial and cervical area. It's the most extensive distribution of any cranial nerve. It has a major role in the control of cardiac, pulmonary, digestive, and urinary function. It's involved in swallowing, speech, and regulation of the viscera, and damage to it can cause hoarseness or loss of voice, impaired swallowing, and you will die if both of the vagal nerves are cut. And the reason for this is because of its impact on heartbeat, respiration, and liver and kidney function. It's the inputs to the viscera that are keeping you alive. The vagus nerve is involved with autonomic input to each of these organs. This is information that controls their function without our conscious awareness. And again, you can see all the organs that the vagus nerve has a role in innervating: the lungs, the heart, the liver, the stomach, the spleen, the kidneys, and the lower GI. In an unusual break from what we think of as voluntary and involuntary nerve inputs, it has been documented that certain individuals can exercise voluntary control over vagus nerve output. The most startling example would be certain Tibetan monks who are able to consciously raise or lower their heart rate as a result of voluntary control exercised over the vagus nerve. Most individuals are unable to accomplish this. It's thought to be a hallmark of a deep contemplative state, and obviously an indicator of a greater degree of control over bodily function than most normal individuals have. So if you want to Google that and look up vagus nerve in Tibetan monks, you'll see some pretty amazing examples of voluntary control exercised over what was once thought to be purely involuntary bodily function. Okay, the spinal accessory nerve is also an unusual nerve. It innervates muscles involved in swallowing, as well as muscles involved in head, neck, and shoulder movement. You can see that it actually exits the jugular foramina on either side of the spinal cord, and basically shares a common exit with the vagus nerve, except that the spinal accessory stops much shorter than the vagus nerve does, and innervates the muscles of the sternocleidomastoid and the trapezius, and is again one of the more rostral nerves in the cranial nerve countdown. Damage causes impaired head, neck, and shoulder movement, and your body will tend to turn towards the injured side. If we look at the final cranial nerve 12, the hypoglossal, it's involved in tongue movements for speech, food manipulation, and swallowing. If both of these are damaged, you cannot protrude your tongue. If one side is damaged, the tongue will deviate towards the injured side, and this is an example of what we term ipsilateral atrophy. Remember, atrophy is degeneration due to disuse, and ipsilateral means on the same side as the injury. Notice that the nerve uh, exits from the brainstem through the hypoglossal canal and innervates the intrinsic and extrinsic muscles of the tongue, and as a result, it's primarily a motor nerve, although again, it is receiving sensory information from muscle spindles that are resident in each of these skeletal muscle uh, tissue masses. Okay, from it's getting proprioceptive information from the extrinsic and intrinsic muscles of the tongue, and this allows the nerve um, to adjust the motor output to so that the muscle doesn't become overly stretched or overly contracted, and we avoid, as a result, muscle damage. And if you want a sort of a brief refresher on why that's important, what you should do is go back and look at the patellar tendon reflex and how that works, and that'll give you some idea of um, the mechanism of the stretched tendon reflex and the utility of that reflex in allowing us to avoid muscle damage. And that's it for chapter 14. So what I'll have posted for you in addition to this video podcast is a practice lecture test that will address aspects of chapter 12, 13, and 14 that are going to be covered on your first lecture test. I'll post that along with a key, and what I would like you to do is take the practice test and see how you do, and if you have any questions about specific portions of the exam, just email me or find me after class or during lab, and we can address any problems